Chapter 9, Selection of Microbial Control Methods. The learning objectives for this topic are: list factors to consider in selecting a microbial control method, identify the 3 most resistant groups of microbes and explain why they are resistant to many antimicrobial agents, discuss environmental conditions that can influence the effectiveness of antimicrobial agents, compare and contrast 4 methods used to measure the effectiveness of disinfectants and antiseptics. In this topic, we will be covering factors affecting the efficacy of antimicrobial methods and methods for evaluating disinfectants and antiseptics. There are a number of things to consider when you're selecting a microbial control method for a particular situation. These factors are, first off, site to be treated. If you're going to be looking at treating living tissue, humans or animals, you're going to want different characteristics than if you're going to be treating non-living hard surfaces. What you want to consider is invasiveness. You don't have to worry about invasiveness on hard surfaces, but you do worry about invasiveness with humans and animals. Also, toxicity. You can have something that is far more toxic that you're using on a hard surface than if you're going to be using it on living tissue. Next, let's talk about item number 3, because item number 2, resistance of microorganisms, I think needs an entire slide all to itself. So let's talk about environmental conditions. Temperature will affect the operation of most microbial control methods, unless the control method is temperature in and of itself. Think of it this way. In the previous chapter, we talked about microbes, how they have optimum temperatures because their enzymes and their structural proteins and their lipids and their membranes are all operating at optimal temperature or optimal vibrational energies. So when we're talking about control methods, especially chemical control methods, you have kind of the same thing going on. You have optimal temperatures for or agents that are going to be attacking proteins and enzymes or busting up cell membranes. So it makes sense that temperature is an important consideration. Now, for chemical control methods, generally speaking, they're tested at room temperature. Why? Because that's where temperature you're going to be using it at. pH is another consideration. Just like pH affects structural proteins and etc. in cells, it also affects especially chemical control method because it may change the activity of the chemical that we're working on. Here's another way to think of it. Something that has passed testing on a clean surface may behave differently when you're cleaning up acidic vomit. It makes sense. And that leads into my next thing, bioburden. Bioburden is any soiling of a biological nature. So vomit, blood, saliva, urine, anything that comes from a biological source that may inhibit the action of a control method is called bioburden. Here's another way to think of it. Say you're cooking a cake and you put it in the oven, and what do you do when you reach the time that it says to check it? Well, you put a toothpick in the center and you see if the center is done. Well, do you ever check the edges? No, because the heat is going from the outside in. Well, the same thing happens with temperature sterilization methods. It has to move from the outside to the inside of the bioburden. If you don't have bioburden, if you don't have cake batter, if you want to think of it that way, then of course the center is going to heat up faster, say in an empty cake pan, than say in one that's full of cake. So think of it that way. Now, I've used temperature as an example of how bioburden can affect the efficacy of a control method, but bioburden can affect the efficacy of just about every microbial control method. On to item number 2 of factors affecting efficacy, which is the relative susceptibility of microorganisms. Here is the figure from your book, and it shows most resistant here, most susceptible here. That means that enveloped viruses are easily killed, easily inactivated, since they're not really alive, by microbial control methods. Whereas the only way to inactivate a prion is incineration and a few enzymes that are specific to prions. So I agree with the book here. There's only one area in which I disagree with the book: non-envelope viruses. The book puts them clear down here in with active cells, and personally I think they're up here with the more resistant organisms. So let me tell you Teresa's categories of resistance. Prions are impossible. Like I said, there's only two ways to inactivate them: incineration, in other words, burning it to carbon, breaking up the covalent bonds that holds the protein together, or enzymes that are going to do the same thing. So this is the category that I call impossible to control with microbial control methods. Next are the difficult to control, and I'm going to include non-envelope viruses in that category. Now, I have basis for disagreeing with the book because I used to do disinfectant testing, and we would use a non-envelope virus, and it was right up there with the acid-fast bacteria, the mycobacteria. It's really hard to control with microbial control methods. So these are the difficult ones: bacterial endospores, mycobacteria, cysts of protozoa, and non-envelope viruses. And the way that I remember these is anything that doesn't have a membrane to attack, or a membrane that is hard to attack. So bacterial endospores and cysts of protozoa are resting stages. They've got a water-resistant, heat-resistant coating, waxy coating that protects them. Non-envelope viruses do not have an envelope that can be busted up by some microbial control methods, so you have to go with attacking the proteins of the capsid. Then mycobacteria, we haven't gotten to that point yet. Yay, chapter 11. But they have a waxy coating that protects them, similar to the bacterial endospores and the cysts of the protozoa. The next group are the moderately difficult. So that would be active stage protozoa, most gram-negative bacteria, most gram-positive bacteria. In practice, there's not that much difference between these two. Fungi. So basically, I just remember this is everything except for the hard stuff, the impossible, and the really easy envelope viruses, because they have a lipid membrane and they have receptors sticking out of that lipid membrane that they use to attach to host cells. Well, if you bust up that lipid membrane, the virus no longer has the receptors, it can't get into cells. You've inactivated it. So, this is how I want you to remember the resistance of microbes. Let's get back to how this affects your selection of microbial control methods. If you're trying to kill, say, fungi in a public gym, athlete's foot on the tile floors, you're going to want to use something that is a mid-level disinfectant or control method. If you're trying to get rid of bacterial endospores, say anthrax endospores that have been put through the mail and have now contaminated several post offices in the DC area, I'm thinking what happened in 2001, then you're going to want to use higher level microbial control methods than you would to get rid of the athlete's foot fungus. Now, before we move on, enveloped viruses. If we're talking about some envelope viruses, it doesn't take much to kill them. But there are times when we act like envelope viruses are the next to prions. And that's because there are some of them that are completely fatal. You always die from them, and we don't have a cure. Specifically, HIV and the hepatitis B and C viruses. Hepatitis B, we do have a preventative measure, but we don't have a vaccine for Hep C, and we don't have a vaccine yet for HIV, although research is looking very good on that point. Until we get a good vaccine for these blood-borne pathogens, even though they're envelope viruses, we treat them like they're endospores, because you just can't risk it. Also, they come protected in bioburden. Blood! The blood helps protect these viruses. So we'll be talking about the bloodborne pathogens recommendations and why we treat them like they're more resistant than they actually are. Next, let's talk about how we evaluate various microbial control methods. In other words, how do we tell whether they're a sanitizing agent, a disinfectant/antiseptic, sterilization agent or method, and through that determining which situations we can use them in and against which microbes. So the first one is the original test, the phenol coefficient. That's where, especially with chemical control methods, we compare the chemical to the original disinfectant, phenol, or carbolic acid. What we do is take chloramine, for example, and if we're comparing that to phenol and its ability to kill Staphylococcus aureus, it is 133 times more effective than phenol in killing this gram-positive bacteria. If we're taking a look at a gram-negative bacterium, Salmonella enterica, it's 100 times more effective than phenol because the numbers have gotten so high. Phenol is alright, but it's not all that effective as a chemical control method. We've got much better stuff, obviously. So the phenol coefficient as a test is pretty much going away. In some situations, we still use it as a control, more to test to make sure that these organisms haven't become more susceptible through being grown in the lab. Microbes, when they're grown in the lab, it's kind of like being on the Riviera. Life is easy. And if you move from Boise to the Riviera in France, then you start to wonder why you need your wool coat. So you get rid of it. Bacteria do the same thing. They kick out genes. Mutations happen. They're not deleterious in the lab, so we have to make sure they're not getting too wimpy. The next method of evaluation of chemical control methods is the use dilution test, and this is the most common test used in the United States. So what we do is we take metal cylinders that are sterile, we call them penny cylinders, and we put it into a broth culture of standard test organisms. So we have 2 gram negatives, 1 gram positive, and lately they've been kicking in an acid fast. Sometimes they'll include a fungus too. Depends upon what use they're testing it for. So then you expose these penny cylinders to disinfectants at different dilutions. Then we expose them for a set time, 10 minutes, and we place them in broth. We take the penny cylinders out, place them in broth, and then we look for turbidity at 48 hours. Here's the idea. You have organisms that are grabbing a hold of these metal cylinders, so it's your surface in miniature. You're looking at different dilutions. So you know that whether you should dilute something, you can dilute things with either bioburden, or if somebody makes a mistake when they're making up the active disinfectant from a concentrate and different things like that. With the broth, we're looking for alive versus everything's been killed, and we leave it for 48 hours in case some of the cells have been injured, but then they recover. Now this is a good test, but personally I see some problems with it. The biggest one is the 10 minutes. I don't know anybody who keeps the surface wet for 10 minutes. 1 minute is about what most people do. They spray it on. If you tell them to wait, they might wait 30 seconds. But that is the standard that the FDA and the EPA use. Now, before we move on from this test, I also wanted to mention that you can modify this test to test in the presence of bioburden. So you're not only exposing the metal cylinders with the test organisms on it to the disinfectant, but the disinfectant mixed with usually 5% horse serum. Lots of blood proteins, lots of good bioburden, and horse serum is fairly inexpensive to get. let's move on to another method. This one, the Kelsey-Sykes capacity test, is more commonly used in Europe. So in this one, we also have test organisms. We have a gram-negative and a gram-positive that we're testing against, medium-level resistant organisms, and we put the organism into the disinfectant. Then we take out samples and put it into a disinfectant inhibitor, and we do this at standardized times, usually 1 minute, 3 minutes, 10 minutes. Then we check for turbidity at 48 hours for the same reason that we check for turbidity at 48 hours, looking at the use dilution test, seeing what organisms can grow if you give them plenty of time. So the main difference of the Kelsey-Sykes capacity test and the use dilution is we're testing time or exposure of the disinfectant to the test organisms instead of dilution of the disinfectant. A test that is becoming very popular is the in-use test. The use dilution test and the Kelsey-Sykes capacity test are both in lab. And as we all know, what we do in development may not be how a product behaves in real life. So the in-use test is an attempt to take a look at how well chemical control methods work in real life. So, for example, we would have a mock-up of a surgery, or sometimes they'll go into a real surgery, dental operatory, etc., and they would take swabs before and after disinfection. Then you take the swabs and you put them in media, and once again you look for growth or lack of growth. With this, you're looking in little crannies. You've got people actually going in and using the product how they normally would, and you get to see, like I said, how it behaves in real life. There's just one problem with this test. We're looking at the ability to recover organisms from these swabs and to collect organisms from the swabs. So for an in-use test, oftentimes they'll take one of these standardized lab test organisms and they'll put a known amount down, and you run positive controls to see how much you can pull off the swab just in the normal course of things. Of course, with the use dilution test and the Kelsey-Sykes capacity test, you're also running controls to make sure the experiment's working right, and so that you can see what log reduction you're getting. Well, that's it for this topic. Next, we'll be talking about actual methods, starting with physical and moving on to chemical control methods.